I was given this brief of trying to look at kind of how my research has been impacted by Epilepsy Ireland funding. And really, when I reflected on it, this support has been key in providing the seed, or one of the seeds, that has led to the Future Neuro Centre that we have today, which I think is a really big opportunity for Ireland understanding more about epilepsy and other neurological conditions. So by way of background, Peter mentioned I'm a geneticist. I moved to Ireland in 2006, which sometimes feels like just yesterday, but I was reflecting on it earlier today, and it's when Italy won the World Cup, and there's been drastic decline in form since then. And it's also when Epilepsy Ireland, or the Irish Epilepsy Association, was known as Brainwave. And at that time, when we think about the genetics of epilepsy, we felt our knowledge was, well, of course, it would have been cutting edge for 2006. We knew about a number of different genes that caused largely familial forms of epilepsy. So these are type of epilepsy that you see a very strong family history of. You might have a sister with epilepsy, or a parent, or perhaps both. So we call these familial epilepsies. And then we knew about 2 or 3 genes that caused what we refer to as encephalopathies. So these are very severe early onset epilepsies where there could be learning disability associated with the condition and developmental delay. But by and large, although we knew about this cluster of, 5 or 10 genes, they represented a tiny proportion of people living with epilepsy. So we knew that genes were playing a role, but it was, we knew very little about the detail of the role that they were playing. So when I moved to Ireland, I was lucky that some others, namely Norman Delante, Colin Doherty, and Mary Fitzsimons, had helped kind of lay the foundation for the work that I was hoping to do. And this was in the context of adult forms of epilepsy. So you're probably familiar with these individuals, Norman and Colin. Well, Norman still is a consultant at Beaumont Hospital, and Colin was working there at the time. They both helped establish a biobank for epilepsy. So a biobank is basically a collection of DNA or biological samples together with information about how a person is responding to epilepsy treatment or the type of epilepsy that they had. So basically at outpatient clinics, so these are kind of weekly clinics at Beaumont Hospital, people with epilepsy were being invited to donate a blood sample and also access to their medical record to help us study and learn more about the genetics of epilepsy. Around the same time, Mary Fitzsimons was she is a pioneer of electronic health records in Ireland. So she, with support from the Health Research Board, was designing the system that is now used on a daily basis at Beaumont Hospital and other hospitals around the country. So it was one of the first electronic health systems in the Irish health network, and it was certainly the first for epilepsy. And that's important for research because when we study the DNA of people with epilepsy, we need to be able to understand what type of epilepsy that they have. So one way to do that is to go and look in the chart. We've all of us who've been treated in a hospital will have a patient chart, right? But these are actually very difficult to access, and obviously they take time to go through because it's all paper-based. You have to find the right piece of information that you're after. Mary's electronic health system changed all that because in theory, you could access very rapidly details on an individual's epilepsy without having to go digging through paper charts. So that helped kind of facilitate our genetic work. Then at the end of 2008, Brainwave at the time put out this call. It was the first of their calls, as Peter alluded to, for research funding. And Mary, Colin, Norman, and I thought we would put in a project around the pharmacogenetic study of chronic refractory epilepsy, right? So what this means is pharmacogenomic is ways in which you can look at your genes and try and infer what treatment an individual should go on based on their genetic profile. And this was in the context of chronic refractory epilepsy. So very, very difficult to treat epilepsy where perhaps an individual has not responded to 3 or more anti-epileptic drugs. So I was digging through the report that we made on this grant, and this is some text that I've lifted straight from it, and I apologize for putting a kind of very wordy slide up, but the bit underlined in red, I think, highlights the key pieces. we managed to recruit through clinics at Beaumont and St. James's over 400 people into our study, which was great. We, with Elijah Chayla's help, so Elijah Chayla, some of you might know him, he's now a consultant neurologist at Limerick, so Elijah was employed on this grant whilst he was training at Beaumont Hospital. So Elijah managed to kind of characterize individual responses to treatment, so we understood how each person in the project had responded to their therapy. We generated extensive genetic profiles on all the individuals in the study, and then we looked to correlate those profiles with the response. And unfortunately, we did not find any predictors, genetic predictors for chronic refractory epilepsy. So that was a disappointment from our perspective. But the next paragraph, I'd put in this little bit of text, which says this grant has helped establish an important resource that can now be integrated into larger international collaborative efforts. And I think in that video, I mentioned the importance of large-scale genetic studies of epilepsy. So basically what happened was around 10 to 15 years ago, the community of geneticists realized that it's really, really difficult to study a disease at one single research site. And the reason for that is you need a lot of people participating in your study to have the power, the study power to be able to find the effects that we're looking for in our genes. So as a result of that, the community of geneticists quickly realized that you need to collaborate within a country, within an institute, within a country, and also cross-border. So as a result of that, in epilepsy as well as in many other conditions, these genetic consortia started to emerge. And the basic idea is, say if we had recruited 400 people with epilepsy into our study, and someone in Australia was doing something similar, a genetic study of epilepsy there, we'd look to merge our results to see if in combination you start to see like signals emerging from the data. So in this context, and using the data that we produced through what at the time was the brainwave funding, we've helped contribute to a number of important discoveries, many of which have started to impact on the clinic. So for example, this work helped the community identify, or this Irish data set, with many others, I should stress, helped the community of geneticists identify that certain deletions or duplications in our DNA, which we refer to as CNVs, or copy number variants, are important risk factors for common forms of epilepsy. So up to this point, we appreciated that very large changes, like millions of bases in size that you can see under a microscope, can be involved in really, really rare forms of epilepsy. But this study, this project and others show that these CNVs are contributing to more common forms of epilepsy. We showed that genes that are important in our immune system, so specifically the HLA, are predictive of very severe skin reactions to carbamazepine. So this is conditions such as Stevens-Johnson syndrome or toxic epidermal necrolysis. So these are like rare but extremely severe and life-threatening adverse reactions to carbamazepine. So this data set helped illustrate or helped identify these genetic predictors that are now on the label. If any of you are using Tegretol, if you look at the label, you'll see these warnings for certain genetic types that, you know, if an individual carries that type, they should not be taking Tegretol. This data set helped position Norman and the kind of Irish genetics community to participate in a large project called Epi4K, which published a landmark project in 2013 showing that extremely rare, what we refer to as new or de novo mutations, so these are mutations that we each carry, but our parents did not carry, right? So these mutations were created in the gametes that made up our first cell. So these mutations, we now appreciate, can sometimes cause very severe early onset epilepsies. And we now understand that upwards of 35 to 40% of people with an encephalopathy have an identifiable genetic cause. And this is an important diagnostic tool that's used now in the clinic. In 2014, this same data set contributed to a large genetic study of the common epilepsies that pointed to a number of different risk factors across the genome. Later, it helped contribute to the finding that ultra-rare mutations, not dissimilar to the ones that cause encephalopathies, they are also contributing to common forms of epilepsy. And then about a year ago, again, same data set contributed to a study that showed or that identified genetic predictors for an adverse reaction in rash to the drug phenytoin. All right, and most recently, just earlier, or I think it was late December of last year, we published a study, another large international consortia, that identified multiple new predictors, genetic predictors for the common epilepsy, and also critically pointed to potential drugs that could be repurposed for use in epilepsy. So the genetic data was potentially guiding, or identifying new drugs that could be used for epilepsy. And I should say that despite all of this progress that has been made, so this is the same slide I showed you earlier, where around the time Italy won the World Cup, we knew about 9 or 10 genes. And then over the last, 9 odd years, there's been an explosion in genes that have been identified for the epilepsies. So that explosion has been driven by collaboration, firstly, and secondly, the development of so-called next-generation sequencing. So it's a faster, much cheaper way of sequencing our DNA that gives us much more power to identify genetic causes for the epilepsy. Now, one of the things I'm disappointed to say as a geneticist is that despite all of these new genes being discovered, very little progress in reality has been made in terms of how you change treatment, right? So as geneticists, I think we're helping explain why a person has epilepsy, but the real challenge now for the community, and hopefully something we can do through Future Neuro, is help identify, new treatments that can be used whether an individual carries a specific identifiable mutation that alone is causing their epilepsy, or as is the case with the majority of people with epilepsy, it's a contribution of nature and nurture, genes and environment, where there's kind of much more complex signatures that need to be corrected for at a biological level. So Linda kind of asked me to talk about the challenges that we've faced in doing this kind of research. So firstly, recruitment in a busy public clinic is very, very challenging, right? Like whether it's the physical space where an individual can recruit, or having the resources to be able to employ someone to recruit, or even pulling someone away from the clinic where an individual obviously doesn't want to miss their consultancy. So when you take them aside to tell them about a research study, that can be a challenge. So recruitment is a challenge. GDPR and the health research regulations are a challenge. So I think we're all familiar with GDPR, and it's impacted everyone at some level. So it's also impacting how we conduct research. I think generally speaking, it's a good thing, but we need to have the right kind of infrastructure in place to be able to apply these regulations to the benefit of everybody. One of the challenges that we're also having, and that we're recognizing more and more, is that we need to get people with epilepsy and people affected by epilepsy more involved in our studies. So that goes beyond just asking someone for a blood sample, to actually having people involved in how we design our studies, how we govern them, how we run them, how we present our patient information leaflets, and even the papers that we write. So we're really keen to have people affected by epilepsies involved in our work, and hopefully you'll hear more from Future Neuro on that front. Research ultimately, right, is about creating a strong environment, right? So if you think in your heads of the best institutes or the most famous ones for research in the world, you probably think of Oxford and Cambridge. It's not that the people who go to Oxford and Cambridge are any different in their makeup. It's just the environment that's been created there is exceptional because it's had many, many years of kind of funding, attracting the best people, and creating the most competitive environment. And that's what I think we are starting to do now in Ireland. And the funding of the Future Neuro Centre kind of recognizes that and gives us an opportunity to build on that and create the right environment, or the best possible environment. When you do that, you attract the best minds to study and help with epilepsy. Because again, research is in effect a competition. You're trying to attract people who are well-trained as scientists, who've been through good environments, to study the condition that you feel passionately about, which in our case is epilepsy. How do we get them to study epilepsy and not schizophrenia? That's about creating really strong environments. And in that context, the funding from Epilepsy Ireland has been critical because it's provided, in many ways, the seed, whether it's the project I'm describing or other projects, it gave the seed to help us kind of create and generate the momentum that comes with Future Neuro. So just for the last slide or two, I wanted to say a couple of words on Future Neuro. So it's funded by Science Foundation Ireland, which is another science funder. It's kind of one of the two, or one of the three main state agencies who fund research in Ireland. Their director, a number of years ago, came up with a strategy for funding research in Ireland. And the logic was, rather than trying to fund individuals, let's look for areas of strength across Ireland, right? So they allowed the research community to prepare bids that were cross-institutional, but you had to make the case that we were world-class in this particular area. So that could be, renewable energies or perhaps the production of milk. And in our case, it was, we're really good at understanding neurological diseases, in particular epilepsy and ALS. So we gathered, or David Henschel, who led this bid, gathered a number of investigators from NUIG in Galway, from DCU, from Trinity, from UCD, from RCSI in Dublin, and other collaborators and investigators around this group. And we prepared this bid to kind of tackle epilepsy and other neurological disorders or diseases across three different broad themes. We wanted to improve diagnosis, right? So better genetic diagnostics in the clinic, better biomarkers, so blood-based biomarkers that might predict, when a person is going to have a seizure, or, predict what therapy might be most applicable. We wanted improved therapeutics, so David and his team here at RCSI, and you'll hear more from Christina about these novel ways of treating epilepsy, of correcting, changes in the expression of multiple different genes in parallel, which we hope and we think could be the next generation of anti-epileptic drugs. And also we wanted this work to be e-health enabled, to build on the work of Mary Fitzsimons, Colin Doherty and Norman in developing electronic health records for epilepsy in Ireland. So using those data sets to better empower our research, whether it's understanding or better understanding clinical decisions in the clinic or treatment-related decisions in the clinic, or correlating types of epilepsy with the genetic work that we are doing. So I'm going to pause there. In particular, I'd like to kind of recognize all the people in the Future Neuro Center who've helped us kind of realize or kind of get us to this particular point. But most importantly, I wanted to acknowledge the people with epilepsy and affected by epilepsy who've donated their DNA and their data to our studies over the last decade. So I'll pause there, and if there's time, I don't know if we're having questions now. Thank you.